Well, thank you very much, and it's a pleasure to be here and present the work that we do in the lab. And in a more general level, what we are interested in the lab is how the genome is organized into gene circuits and how these gene circuits give rise to the huge variety of cells that we have in tissues. However, we know that this is not such a simple process where the information goes from the genome to the cells to the tissues, but also the information from tissues feed back into these gene circuits, and this has an impact on the cellular identity. So the main question that we have in the lab is how does the microenvironment is rewiring these gene circuits and how this has an impact on cellular identity. So our favorite model of studying this interaction between the microenvironment and the cellular identity is the immune system. The immune cells develop in lymphoid organs, but then sit tissues where they are influenced by their microenvironment, and that gives the final identity of this cell type. So to address these questions about the cellular microenvironment and the cellular identity, we use an iterative wet drive lab framework, where first we use the cellular atlasing technologies that were just described. So it was fantastic to go after this talk to profile cells in their native context of the tissues, and we use single cell and spatial transcriptomics and other modalities, technologies. Then we use and also develop computational tools to extract biological information from this huge amount of data, and also making predictions that then we validate using in vitro models that allow us to make perturbations at scale. And I say that this is an iterative framework because actually our readouts from perturbations are also single cell readouts that will then get integrated into the initial profiling, and ultimately this allows us to make better predictions that then go validated really in a very clever iterative approach. So one of the models where we have been looking for these tissue-resident immune cells have been the reproductive tissues. The reproductive tissues are really also quite kind of interesting to study this interaction between the cell and the environment, as they are quite dynamic and influenced by the sex hormones. And these, again, allow us to have a new angle for this exploration between the microenvironment and the cellular identity. Reproductive organs are essential for the perpetuation of the species, as they enable for the genetic information to go from generation to generation, but actually they also have a crucial role on women's health. So a clear example for that is endometriosis, where uterine-like cells are grown outside the uterus, and that causes a painful, really disorder that affects 10% of women in reproductive age. So I'm sure all of us know someone with endometriosis. So the fact of reproductive biology really is that it has been neglected, and actually we knew very little about the molecular and cellular kind of composition of the reproductive tissues, and that really has delayed any therapeutic strategy that we can think about. And that's really quite impactful if we compare what we know about other mucosal tissues. So our team has been leading on this single-cell female reproductive genomics to really give a new angle on the molecular and cellular mechanisms for reproductive tissues. with the idea to create a universal code, like a Rosetta Stone, where we can integrate previous information that we knew about the tissue, and with all of these extract new biological information, kind of creating gold standards that everyone can use and incorporate as part of their pipelines. So we have been really, at the beginning, focusing on mapping healthy tissues, and not only the immune cells, but also the non-immune cells, because if we want to understand what the immune system is doing, we also need to understand what the other part is doing. And we have been looking on purpose across the lifespan, because the reproductive tissues are quite dynamic across the lifespan. They are developed in uterus, but they are not functional until after puberty, and then they go to massive remodeling during pregnancies. So we have been mapping all this dynamic of reproductive tissues, but for today, I'm going to focus on our work on the adult endometrium. So what is the endometrium? The endometrium is a mucosal layer, and in particular the mucosal layer of the uterus. And as in many other mucosal tissues that we have in the body, is enriched in immune cells. And one uniqueness of this mucosal layer is that goes into cycles of regeneration that is mediated by the two hormones, estrogen and progesterone. So actually, this regeneration is a perfect regeneration. It happens without a scar, and it really involves the layering of the tissue, the regeneration of all the epithelium, the fibroblasts, and all the arteries. And so really are really complex tissues that gets regenerated every month. And I will be talking about the work that two members of my team did, Luz Garcia Alonso and Magda Marekova. So the way we went into the tissue was to take samples from multiple individuals, because it's quite dynamic, so we needed hundreds of them, and also introducing data from others that was published. And that is like having multiple screenshots, and we really managed to cleverly find computational ways to integrate the data by also having other multimodal readouts. And one of the readouts that was crucial for us to have some anchors and give identity to these clusters was the spatial information. So I'm going to give an example so you understand what I mean. Well, I don't think this is working, but there in blue we have all the different colors of the epithelial cells, meaning each color will be a cluster with unique markers. And for example, if we start with this number 1, we found that this was consistently being mapped to this layer, which is the end of the endometrium, and then will come the myometrium. And this is the layer that doesn't get shed during the menstrual cycle. And also this population expressed a lot of progenitor markers, which allow us to give the identity of potential progenitor of the epithelium of the endometrium. Similarly, we also identify another cluster here, number 4, where actually it was mapped into the lumen, here in blue, which allow us to, and also express some progenitors, which allow us to give the identity of a progenitor luminal marker. And doing this with all the different clusters that we have in this map, then we can start getting models of what's happening in the tissue. So this is a mucosal tissue where you have different glands, and we could identify how the different populations from the progenitor to the different glandular states emerge, and how they change in both space and time, and similarly for the ones in the lumen, as well as some ciliated populations that also emerge in the lumen and that enable to really spread the secretions that are produced in the glands. So in addition to the epithelial cells, which are the structural cells of the tissues surrounding them, we also have the stromal cells, which are specialized fibroblasts. And sometimes I like to call it fibroblasts because you don't have fibrosis in the tissue. It's a scarless differentiation. And basically, again, we could identify the different subsets of these stromal cells, how they transform to each other, and how they really express different markers in different stages of the menstrual cycle, which you can appreciate is quite dynamic and will change completely every 4 and 5 days. So having these fibroblasts or specialized fibroblasts together with the epithelium is quite interesting, because then we can get understanding of what are the signals that are happening in the tissue and that are driving the final identity of the epithelial cells, which are the structural cells. So how we do that? So for that, we use a computational tool called CellPhoneDB, which I developed when I was a postdoc in Sarah Teigman's team, and then my team has developed it further. And basically, this tool consists in 2 things. First, a repository of ligand receptors and their interactions, and we consider that the majority of ligands and receptors are multimers, and that determines the specificity. We also consider non-protein receptors interactions by reconstructing the enzymes production reaction. And on top of this database of ligand receptors interactions, we also have a statistical method that looks for specific ligand receptors interactions in a given single cell data set. We can give layers of confidence for these interactions by, for example, also looking at the downstream signaling, so transcription factors that are active or not active, for example, looking at the multi-omic data with attack data. And if we have a spatial data, we can also start looking for interactions that are happening close by as interactions that are most likely more relevant or more likely to happen. And with this, we can get some predictions to prioritize the ligands and receptors interactions that are happening in the tissue. And that's actually what we did in our case of this mucosal tissue, the endometrium. So examples of interactions that we could see here and signaling happening was, for example, TGF-beta inhibition happening from the lumen, or Wnt inhibition happening around the glands, and with this inhibition being supported by the surrounding stroma. And also we found that the epithelial cells themselves seems to control different gradients of notch, becoming notch activation high in the glands. And we really strongly felt that that had an impact on the epithelial identity, based also on the transcription factors that were activated in the epithelial cells as they differentiated. But of course, these are human samples, and we cannot demonstrate that in vivo. So instead, we went into the organoids. But by then, the organoids were relatively new. This was in 2020, more or less. And we needed to find a way to benchmark them. And because we had these atlases of the tissue, there was a unique opportunity to benchmark these in vitro data against in vivo reference atlases. So these organoids, you grow them first without hormones, but then you can add hormones with estrogen, recapitulating the proliferative phase of the menstrual cycle, and we found that gave rise to clusters and cellular populations of the epithelium that you find in the proliferative phase. And then if you expose them to progesterone, they will start producing more secretory cells and glandular cells that you also find in vivo. So in our hands, we could demonstrate quantitatively that the organoids were a very good model for us to look into pathways that are important for epithelial differentiation. They were only recapitulating early kind of secretory phase, but for the purpose of our studies, which was really to show the signals relevant for this epithelial identity, they were good. So what really we wanted to explore here was the signals. On one hand, all these Wnt inhibition that we found that the stromal cells are giving to the epithelial glandular cells, and that this is important for their identity, and also the notch activation that happens in the glands, which is supported by the epithelial interactions. So again, we went into the organoids, and this time what we did was to grow the organoids in the presence of notch and Wnt inhibition. And what we found is that if we added notch inhibition, you have a lot of ciliated cells, and you have very few secretions, as you can see by the staining of PAP. And if you compare it with controls, I'm sorry that the laser is not really working. And that really recapitulates what happens in the lumen, where you have a lot of cilia and you don't have that many cells that are secretory. On the opposite, when we added Wnt inhibition, which is what will happen in the glands, where you have the glands surrounded by these specialized fibroblasts that provide this Wnt inhibition, then you have very few ciliated cells, but you have a lot of secretion, as happens in the in vivo tissue. Of course, to this point, we didn't know if the secretions was because we have cells that are better at producing things, or it was just that we have more cells producing things. So that's where we applied single-cell transcriptomics, and basically we could see that now we have more secretory cells. I cannot show you the plots here, you need to trust me, but also we have a new cluster for secretory epithelial cells, and actually this new cluster that appears with Wnt inhibition express higher levels of these secretory markers, really indicating that this Wnt inhibition will not only help on producing more of these epithelial secretory cells, but also produce secretory cells that are better at secreting. Good, so the way I see all of this is that with all these atlases that we are generating, we are decoding the rules for cellular identity. We are identifying the distinct cell types and cell subsets, the markers they express, but also gaining insights into the molecular pathways that we need to produce them. And this is really creating a rich encyclopedia of information that then we can utilize for different things. And here I want to point out how our vision is to use them to generate as recipes for really generating better models in a dish. So, for example, we have all the information or all the ingredients in which providing the ligands that we can really supply to the media, or the transcription factors when they are expressed and when they need to be expressed, for example, across a regeneration or developmental process. But also with all the spatial information, and I am not showing much images, but we are also starting to see the different gradients, and for example, with the Wnt and notch, how there are different gradients, and how this is essential to generate the full structure that we need in the tissue. And I want to give you some insights in how are we really going into these gradients and how we can transform them into better in vitro models, also considering all the advances on microfluidics methods. So for this, for example, the current organoids, they have 3D structures, and under stimulation with the hormones, they can become secretory, which is great. However, what we are missing in the tissue is to have the proper glandular structure that you will see in the tissue. And of course, that's relevant if one wants to, for example, recapitulate anything related to implantation. So with this, what we are working together with engineers, with Linda Griffith at the MIT, is actually to build microfluidic systems where we can create these gradients and we inform this, all this information based on our information that we have in the atlases. And it's very preliminary data, and I'm not showing here, but we are starting to see the emergence of these glands when we create the gradients, which is very exciting. Good, so I told you how my vision is to look at the tissue-resident immune cells, and I am an immunologist by training, and this tissue is full of immune cells, and in particular, it has a huge abundance of macrophages, and also it does make sense to look for the immune cells, as a lot of reproductive disorders, including endometriosis, they are inflammatory disorders. So here, again, following our strategy to look at the interactions within the tissue and the ligand receptors, we could also infer function for the uterine macrophages, not only defining their identity, but also really pointing towards an important role for the regeneration of the tissue itself. So they express, we see that they are recruited during menstruation, where the physical damage and the tissue is becoming destroyed, and then start producing a lot of factors that are important for wound healing. And then after, as the regeneration goes, then start decreasing inflammation, which is actually relevant for the scarless differentiation of the endometrium, which needs to happen without inflammation. In addition, again, by looking at the interactions and where they are located, we could see that there are a lot of ligand receptors interactions that are related to angiogenesis, so most likely also being involved in the proper regeneration of or angiogenesis in the process and the building of these spiral arteries that you have in the tissue. Good, so we also wanted to look a bit further of what happens in the context of disease and really considering all the cells that we have in the tissues. And for that, we moved into endometriosis, which really, as I mentioned before, it's quite common, happens in 1 of 10 of those born with a uterus, and really is characterized by the presence of cells resembling the endometrium, but growing outside the endometrium. And this really affects the quality of life of women with a lot of pain, and it's really an inhabilitating disorder. And it really still takes more than 8 years to be diagnosed, with the final diagnosis requiring some kind of imaging or even surgery, and there is no efficient treatment. Women are put in too often into hormonal treatment that sometimes induce menopause, which is not ideal, and also it doesn't work in all the cases. And particularly to start looking at this disease, and this is what I'm going to present here, we were not looking into the lesions, so where the tissue grow. We were just looking at the uterus from those people with and without endometriosis, because actually there is evidence that something is altered in the uterus itself, because those women are also linked to infertility and a lot of menstrual pain. So looking at the data, single cell data from women with and without endometriosis, what we could see is that actually in endometriosis, as you can see here, there was an increase on macrophages, and in particular those macrophages that are inflammatory, and each dot doesn't represent a cell, it's a neighborhood, so to avoid confusion. And also we did see a decrease on the fibroblasts that are responding to hormones, showing that they are less able to respond to hormones. So when looking at the genes, what we could see is that these macrophages, instead of dampening the inflammation across the menstrual cycle, as it happens in the normal uterus, in endometriosis, there is a sustained inflammation that could explain some of the phenotype that we see in those women. So the other thing that we wanted to know is to gain more insights into who is expressing the genetic variants that have been described for endometriosis, and see if it's also the macrophages that are expressing those. Basically, what we did here was to cross our single cell data together with GWAS for endometriosis, and what we found out is that it's actually, again, the macrophages together with these fibroblasts responding the hormones, the cell types that are expressing genetic or genes that are linked to these endometriosis risk loci. And to me, this is also like kind of change on how we see the disease, because we historically have been thinking about it by targeting the epithelial cells that are the structural cells, but here instead what we are saying is that we should look at the microenvironment, because that is what will, in a way, because communicates with the epithelial cells, will be most likely to be the cause of the disease. So really to conclude a bit of the work we have been doing on the macrophage side, we have been seeing that macrophages are crucial for the regeneration of the tissue in terms of they contribute to angiogenesis, they are crucial for tissue repair, but some of these monocyte-derived macrophages are altered in endometriosis. They are sustaining inflammation. They also express a lot of these GWAS genes, and I didn't go into our work during pregnancy, but we also see that these uterine macrophages are crucial for implantation and kind of placentation itself. And really pointing out towards the importance of macrophages in reproductive biology, while still there is a lot of work that we are doing in terms of how they behave in the context of other diseases or even in the lesions themselves from endometriosis. Of course, all of these are predictions that we get from the data, and to ultimately get a bit more insight into the causality of these, we need to establish better or more complex in vitro models that go beyond the epithelium. And for that, we have been setting up models where we have the organoids. They are dark because they are full of secretions, not because they are dead. So these organoids are together with surrounding fibroblasts, and this way making a system that is more complex and that recapitulate the interactions that also happens in the tissue. We are also able to introduce macrophages to the system. They are the small dots that you can appreciate probably there, in a way that we can then start having a system that we can then start perturbing at the scale and really feeding back and forth with the in vivo data. So just to conclude, I started my talk into how our team is interested on cell circuits and how the microenvironment is affecting these cell circuits. And at the present, the way we have been answering those questions have been to look at tissue modeling using in vivo tools. And here for this talk, I focused on our work in reproductive tissues. And also we have been utilizing this information to benchmark current in vitro models, and even making some improvements for them to resemble what we have in vivo in a better way. As we move in the future, we are keen to also start looking at the disease mechanisms in more depth, both by profiling, deep profiling specific cohorts, but also going into the in vitro modeling and perturbations, not only in reproductive tissues, but also some other inflammatory disorders in general. And we have some good collaborations with Sarah Techman of the gut organoids, also with Mushanifah on skin organoids, and of course bringing all the AI tools and this kind of loop of the lab in a loop approach to go between in vivo and in vitro smoothly, also collaborating with Molot Hoyahi at the Sanger Institute. And with this, I just want to finish by thanking my team members, and particularly the work I discussed, led by Luz Garcia Alonso and Magda Marekova, our collaborators and funders, and yeah, happy to take questions. Thank you.